So this is the first of a 7-video series where we're going to talk about protein structure and protein function. And I thought rather than going into the details of protein structure first, I'd instead start with some function so that we have context when we start saying, oh, look, here's an alpha helix and this is why it's important. So let's dig into that. So biochemists have this fundamental paradigm that protein structure or protein structures determines protein function. And I wanted to illustrate that with 4 different proteins that have 4 different functions. So let's just jump straight into these. So the first protein I want to highlight is albumin. This is a protein that is currently floating around in your bloodstream. Its 3-dimensional structure is shown here, where spheres are different atoms, where they're colored by their atom type. So oxygens are red, nitrogens are blue, etc. And it might not be obvious when you look at it, but it's decorated with a lot of oxygens and nitrogens and hydrogens that are polar on its surface, which means this thing dissolves really, really well in water. So it's a very water-soluble protein. What's it doing in your bloodstream? Well, we can see that if we erase the albumin and expose its cargo. And its cargo is fatty acids. Fatty acids, greasy hydrophobic molecules, they tend to stick to each other and they tend to stick to the walls of your blood vessels. Turns out things like arterial plaques are in part things like fatty acids sticking to blood vessels inappropriately. So albumin is basically a shell that's protecting us from the fatty acids and protecting the fatty acids from us so that it can deliver it, say, to fuel something later. It achieves this because it's got specific little pockets on it that bind to fatty acids. So in this view, what I'm doing is I've highlighted the fatty acids as yellow, the carbons are yellow, and hopefully what you can appreciate is there's a nice little pocket where that fatty acid is sitting away from water inside the albumin to be delivered safely through the bloodstream. This illustrates one of the fundamental ways that what biochemists mean when they talk about structure determining function is that a protein tends to bind to specific partners by having complementary sites. Those sites in albumin, they bind to albumin, but not to other stuff, which means albumin can specifically transport fatty acids and safely without running into other things. So if we say protein structure determines function, one way is by having complementary sites. Another example is glucose transporters. So as you are probably aware, glucose is a molecule that we all like. It's a sugar. There are certain blood cell types that specifically require glucose. For example, red blood cells are addicted to glucose as their fuel. But the problem is glucose, say, in the bloodstream can't enter a cell very easily because it can't pass through the bilayer very easily. It's polar, it's sort of big, so it just doesn't like crossing a hydrophobic bilayer. To overcome this, our cells make proteins that specifically transport glucose and not other molecules through the bilayer. So I've shown that now. Hopefully you can sort of see there's something purple moving around in the background. I'm going to now slice through the bilayer, slice through that molecule like an apple so we can see what's going on inside. And hopefully what you can see here is there's actually a channel that is opening and closing in the middle of this molecule. And if you'll notice here in this yellow thing in the middle, that's actually a glucose molecule. This might be easier to see actually if we're going to change our view a little bit. So now I'm going to zoom out. Now we're looking at the same picture we had before, but now it's at the bottom. But we're now looking in from the outside and from the inside. And hopefully what you can appreciate is that the glucose is basically exposed to the outside here. And then when the channel closes, it's now exposed to the inside here. What that allows the glucose molecule to do is diffuse from outside into inside. Even cooler, that pocket, the pocket only opens and closes when a glucose binds. So when glucose binds, it triggers the conformational change, which allows it to specifically transport glucose and not other molecules through the bilayer. So we saw with albumin that we get specificity, that proteins determines function by having specific binding sites. What we're seeing with this glucose transporter is that you add a specific binding site for glucose to changing between structures, and now the multiple structures are determining the function, right? So the transporter allows glucose to bind and move across the bilayer. Okay, so our third example is actin. So actin is a super cool molecule that makes branch networks as part of our cytoskeleton. So what I'm showing here is the structure of actin in sort of this nice painted form. And it can form these stacked arrays of molecules and also form branched molecules when you have an adapter protein. And I'm actually going to show this in person or whatever using Legos. So I've got a Lego brick here, and we can think about actin is it's got a complementary Lego top side and a complementary non-top side. And what actin does is it can assemble end to end, like bricks of Legos. And then occasionally we get a special molecule called an ARP23 that comes in. It's that red guy. And the red guy can continue to allow things to stick on top, just like normal. So it's kind of like a normal actin. But it also has a complementary side where now things can branch off and go in another direction. So we can continually stick more actins on. We can make our filaments come out like this. Whoops, I'm breaking my actin filament. Okay, so again, actin filaments are in gray, so they're allowing it to polymerize and make a chain, and then the red things they're causing it to break. That's an ARP23. So now if we go back and we look at the molecular structures, hopefully you can see that. These are the gray bricks. They form stacked arrays. That's the gray stack. And then ARP23s can make a branched filament there. So looking at my tiny picture at the bottom, we've got that there. Okay, so it can assemble into these things. Turns out we can actually watch this in real life. So this is actually from a colleague of mine at the University of Oregon, out of his lab, Brad Nolan. What he actually did is fluorescently labeled actin and then watched it on a microscope. And hopefully what you see is you can see these growing, beautiful branched patterns. That's actually actin assembling into these networks. This allows for things like migratory cells to sort of push out their membrane and crawl just by polymerizing actin in these branched networks. It also allows other sorts of large-scale motion. So this is a really fun example. The blue cells are bacterial cells that have infected a eukaryotic cell. The pink labeling is actually fluorescently labeled actin. And what you might be able to see is those blue cells are moving across the pink cell. And what they're doing is they're actually polymerizing actin behind themselves. They're like throwing actin out the back. They're stealing it from the host so they can move through the cell. So what are we seeing here? What we're seeing is that a protein is assembling into a specific complex that allows it to do a specific job, right? The ability of actin to form these arrays, these filaments and these branched filaments allows it to do specific jobs. Okay, one last example, and that is an enzyme, serine protease. And I hesitated to put this in, but I decided it was cool enough to talk through. So let's talk about it. I hesitated because it feels complicated, but it's so cool we have to talk about it anyway. Okay, so serine protease is an enzyme that cuts peptide bonds. So proteins are made out of peptides. They're peptide bonds, and this will basically cuts up proteins. It does so via an active site, which I'm zooming in on right now. And in this active site, what we have are different residues, different amino acids that are in position to do this chopping. So in green, I'm showing the active site. So this is something from the protease, the serine protease. It's going to do something to its substrate, which is here in blue. This is what's going to be cut. What's really fun about this is we're going to see how the specific arrangement of these amino acids in green allows a chemical change to the things in blue. So, okay, what we're going to do first, the amino acids in green are arranged to cut peptide bonds. The histidine, so this amino acid up here, takes a proton from the serine, so it stole that proton. That has now made this serine super reactive. It needs to do something with itself, and what it's going to do is it's actually going to do a nucleophilic attack on that carbonyl. That nucleophilic attack results in a new bond being formed. After that attack, that creates, sorry, I got skip steps here. That causes the attack and then causes this to move down into a tetrahedral intermediate. That tetrahedral intermediate is an unstable, wacky form, but the enzyme stabilizes it through a new hydrogen bond that's formed down here. That speeds this reaction up a lot. This is finally resolved by the breaking of that bond here. So basically this is going to, the negative charge on this intermediate is going to come back here. We're going to break that bond there, and we're going to actually cleave the peptide bond. So, okay, that's super complicated in a sense, but the reason I mention it is because it's so exquisite, right? You've got all these things that are arranged in a very specific way to achieve a specific job. So even if I didn't explain it very well or you didn't follow the organic chemistry for the moment at least, hopefully you can just appreciate that this specific arrangement of amino acids in an enzyme allowed for a very specific functional outcome. And so in addition to these other functions we've talked about, we can say that proteins catalyze specific reactions by having active sites that are arranged in very specific ways. So at this point, I want to just highlight something, and that is my sophomore in high school English teacher would be upset with me because I'm not using the different words, right? She'd say, grab a thesaurus, stop using the word specific. But I chose to use that over and over because that's how proteins work. Proteins bind to specific partners by having complementary sites. Proteins function by changing between specific structures. That was the glucose transporter, open or closed. Proteins assemble into specific protein complexes to do specific jobs. So that was actin, building these beautiful networks in a very specific way. And finally, proteins catalyze specific reactions. The exact arrangement of the amino acids in that serine protease lets it cut a particular set of peptide bonds. Okay, so that's what I got on this idea that protein structure and protein structures determines protein function. In upcoming videos, we're going to talk about where that protein structure comes from, but hopefully this is a helpful starting point or a helpful paradigm as we tackle that new content. So thanks, see you later.